Hello, Gen Bio. Dr. Fry here. This week we're going to be talking about histology and specifically different types of tissue that we can observe under the microscope. So the first slide here is just picturing lots of different types of tissues. So the four major types of tissues are represented by these four pictures. The first here is epithelial tissue, and there's lots of different types of that, which we'll look at. And then there's six different types of connective tissue, three different types of muscular tissue, and then nervous tissue is represented by this fourth picture here. Okay, so we're going to work through all four of those and break them down into different subtypes. So the goal of this lab is to have you be able to identify the different types of tissue had you been able to look at it under a microscope. Okay, so first we're going to talk a little bit about how to make slides from a tissue that's harvested from an animal and to put that into a format that is able to be visualized underneath the microscope. So first off, you have to extract the tissue from the animal. So say you take out nervous tissue, say you take out a brain from a mouse or other model organism. You have to extract it and first preserve it. So this can be done through either physical or chemical mechanisms. So one way to do it is just to do a quick freeze dry the tissue. Another way is to preserve it with chemicals such as formaldehyde. And this basically, the goal is to let it remain in as lifelike a state as possible. So we don't want the cells to break down. We don't want the tissue to have any type of fragmented appearance. We want to preserve it so that we can observe it in its natural state. Okay, and then several steps are applied to the tissue to first dehydrate it and then to fix it, again, to retain its state as close to how it would be in life. Okay, and then the alcohol is removed and it's allowed to be penetrated by paraffin wax. So that's shown in the second picture here. It's the tissue surrounded by a big wax block to keep it solid. And this allows someone to cut the tissue to make very, very thin slices. So once it's in that wax block, it can be sliced without damaging the tissue too much. So you can slice it just like you would basically see at the deli counter. You know, it's basically like a really tiny version of a meat slicer. And that's shown here in this third picture. You can see like each of the little pieces of the wax block are coming off of that meat slicer. That's called a microtome, not a meat slicer. So once you have each of those slices, they're going to be very thin, but they're protected from the paraffin wax. And then you can put that onto a slide, remove the paraffin, and then remove some of the other agents that are used and mount a cover slip with a type of glue called resin. Okay, and then you've got your complete slide. Before you would have mounted the cover slip, if you desired, you could also stain the tissue with either some type of dye that binds directly to a protein or to a part of the cell, or you could apply some antibodies and apply secondary stains to that to allow you to visualize whatever part of the cell you as a researcher would like to see. Could be a particular protein, could be cell membrane component, so that you can see the different parts of your cell best. Okay, and so that's what goes into making a slide that we would then again be looking at if we were in lab this week. We'd be looking at lots of slides that have different types of tissue. So let's get right down to the different types of tissue. Animals have four general tissue types, and they're listed here: epithelial tissue, connective tissue, muscular tissue, and nervous tissue. Tissue itself is basically a compilation of cells, so it's multicellular, and these cells work together to achieve a certain purpose. So they then, tissues, make up different organs in the organism. So tissue and organ, I want to clarify, are not synonymous. I often have students who, when I ask, like on a quiz or on the practical, when we ask for a tissue type, they'll say kidney or skin, something like that. When we want a tissue type, we want one of these 4 general types, and then there's going to be subtypes for each of them as well. So just sort of keep that in mind as we go along. These are present in organs, but they are not the same. They're not synonymous. Okay. All right, so the first one is epithelial tissue. Epithelial tissue is found lining cavities and surfaces in animals. So you'd find it on your skin, lining your digestive tract, lots of other linings, so nasal passages, mouth, like I said, skin, exterior surfaces, okay? So it's gonna line internal cavities or external surfaces, and it has densely packed cells. And it is somewhat unique in that it is avascular. There's no vasculature in the epithelial tissue itself. It does have a polarity to it. There's different sides to the epithelial tissue. So there's an apical and a basal surface to epithelial tissue. The apical surface is what's exposed, either to the external environment or to the internal cavity. And then the basal surface is the opposite side, where it's resting on other layers of cells. So this would be the basal surface of this epithelial cell here. And it's resting on other cells or a basement membrane. So the basement membrane is going to act as a scaffold for which the epithelium can grow, and it also will supply needed nutrients to the cells that themselves are avascular, okay? So moving on to the next slide, we have different types of epithelial tissue, okay? So you should be familiar with the different subtypes and be able to identify those by the end of the lab this week. But they can be broken down. The subtypes are, to start with, either simple, stratified, or pseudostratified. Okay, simple epithelium just means that the epithelium is made up of one single layer of cells. Okay, the cells can be squamous, cuboidal, or columnar. Okay, so if you look at the picture here, the leftmost top picture shows simple squamous epithelium. And what that means, again, is that the word simple indicates that it's just one single layer of cells. The squamous describes the cell morphology. So squamous means like it's a squished appearance. You've got very flattened cells, and the nucleus itself might be the most bulbous part of the cell because the rest of the cell is just so squished that you've got that nucleus taking up more room than the rest of the cell because it's so squished, okay? So that is simple squamous epithelium, okay? Cuboidal, simple cuboidal epithelium is, again, simple as one layer. Cuboidal, again, describes the shape of the cells, and that's shown in the middle picture here. So in cuboidal epithelium, you have a rounder nucleus and a much more large bulbous shape to the cell, like a cube. That's why it's called cuboidal, okay? So you've got squished cells, that's squamous, cuboidal, which look like a cube, or columnar, which again describe the morphology of the cell. So they look like columns. They're thinner on one end, the end that lies on the basement membrane or runs parallel to the basement membrane, and then longer on the perpendicular side of the cell to the basement membrane. Okay, so that is the breakdown for simple epithelium. Let's move down to stratified epithelium. This just means that there's more than one layer to the cells, okay? It's epithelial tissue that has at least 2 layers of cells and often more, okay? You can also see that in different forms as well based on the morphology of the cell. So you can have stratified squamous epithelium. And this means that the top layer of cells, the apical side, not the side that rests on the basement membrane, the top layer has a squamous appearance. It does not mean that all of the cells in that stratified epithelium will have a squamous appearance, just the top layer, okay? See, as you can see in the picture down here, the bottom layer looks like it has cuboidal cells, and that's okay. It can have different types of cells on the basement membrane, but the top layer is, if it's squamous, then we call it squamous stratified epithelium, okay? And then that can also, again, depending on the morphology of the top layer of cells, that can be a cuboidal stratified epithelial tissue or columnar stratified epithelial tissue. And then lastly, we can have a third subtype of epithelial tissue called pseudostratified. So it looks like it's got more than one layer. It looks like it's stratified, but it's not really. It's only one layer of cells. And the reason it looks that way, it appears this way, because there's various heights and positions of the nuclei in pseudostratified. Whereas sometimes this can be confused with simple columnar. Simple columnar epithelium has usually the nuclei at a relatively even level in all of the cells in that layer of epithelial tissue. Whereas in pseudostratified epithelial tissue, it looks like the nuclei are at various heights, various positions in each of those cells, okay? So if I ask you for a type of epithelial tissue, you need to keep in mind how many layers of cells there are and what the shape of the topmost layer of cells is, okay? And that will give you the type of epithelium, okay? So let's move to the next slide. And this picture here, the large one, looks a lot like one of the slides that we have in lab. This is taken from the kidney, and it shows two different types of epithelial tissue, okay? So in the kidney, there's lots of tubules. The kidney processes fluid, which ultimately becomes urine. And in order to do that, it needs to have the fluid pass through a lot of tubules and undergo changes along the way. So all these little tubules that you're seeing, all these little circles in this kidney section are different tubules in the kidney, okay? So if you look here, this circle has bulbous nuclei to the cells and relatively cuboidal shape to the cell itself, right? And if you look, it's got one side exposed to a central cavity, and then the other side is a single layer, okay? There's not another layer of cells beneath that. The other side is resting on a membrane. So this we would then be able to identify as simple cuboidal epithelium based on the fact that there's only one layer of cells and that those cells are cuboidal shaped, okay? And then in this section, we can also see simple squamous epithelium, okay? And I'll point that out right here. So you can see again, these cells look very different. They're also exposed to an internal cavity. It's also one layer, but look how squashed that cell looks. The nucleus is the largest part of that cell, and the rest of it's really flat. So that is an example of simple squamous epithelium, okay? So those are great examples of those two. So let's move to the next slide, and we're going to look at an example of simple columnar epithelium. Okay, so this section is taken from the small intestine, and in this section, you can see a couple different things. There's these big gobs of mucus here, and mucus is obviously very instrumental in the small intestine as well as other parts of your digestive system. But so these cells that secrete the mucus that are interspersed here, those are goblet cells. The rest of the cells that you're seeing here lining the small intestine, the rest of these cells are simple columnar epithelial cells, Okay, so if you look, you've got relatively even positioning of the nuclei. You don't have a lot of nuclei up at this end or in the middle. Most of them are kind of at the same level along this section of epithelium. Okay, so this is one indication that we're looking at simple columnar epithelium. And the only other thing we're going to look at in that slide, like I said, is the goblet cells, which produce mucus interspersed in between the simple columnar epithelium. Okay. All right, let's move to the next slide, where we're going to look at now stratified. So now we've got more than one layer of cells, and we have the top layer looking very squished or squamous. Okay, so all of these cells are one section of epithelium. So these are the basal cells, and the apical cells are squamous. So as you can see, there's lots of different layers of cells, so this means it's stratified. And the fact that the apical cells are squished or squamous means we would then call this stratified squamous epithelium. Okay. All right, let's move to the next slide. Okay, so this one, I do want to compare and contrast with our columnar epithelium that we looked at already. So if you look at this one, this is the tricky one. It's pseudo-stratified columnar epithelium, meaning it looks like there's more than one layer. It looks like it might be stratified, but there's only one layer of cells resting on the basement membrane here. And the way to distinguish this from our simple columnar epithelium is look at how even the cell nuclei positions are in this as compared to our pseudostratified. Look at all the different diverse positions of nuclei here. So this is characteristic of pseudostratified columnar epithelium. This is one layer cells. They're all actually resting on the basement membrane, but the nuclei are kind of mixed up in their positions there. Okay, they're not appearing like they're at the same level in adjacent cells. Okay, all right, so that wraps up the examples that we have for epithelial tissue. That was the first type of tissue that we looked at in animal cells. Now we're looking at connective tissue. And again, there's going to be some subtypes of connective tissue. Okay, 6 in fact. So the types of connective tissue are loose, or sometimes called areolar connective tissue, dense connective tissue, adipose, which is fat, cartilage, you probably know what that is, bone, and blood. Okay, so to varying degrees, connective tissue is pliable. Obviously, cartilage is much more pliable than bone. Okay, all connective tissue has cells, fibers, and what we call ground substance. It's like a mixture that is in between the cells and fibers that makes up the background, the backbone, so to speak, of our connective tissue. Okay, so the first subtype of connective tissue is loose or areolar connective tissue. It can be called loose, sometimes called areolar. There's cells present in there that are either fibroblasts, which make up the fibers, which produce the fibers, and macrophages, which function in defense and immunity. So if you look here at this bottom picture here, the dark purple bulbous structures here, those are the fibroblasts, which are the cells that produce the fibers in loose or areolar connective tissue. These are both pictures of loose connective tissue. This one, I believe, looks a little bit more like the slides that we'll look at, but be familiar with both. So again, the dark purple spheres are fibroblasts. And then there's 2 types of fibers that are present in loose connective tissue. There's the long, thick pink ones. These are collagenous fibers. And then the thin purple or like black black colored ones. Those are elastic fibers, okay? So they're darker and thinner. Okay, and then there's just, like I said, there's a ground substance. It could be kind of liquid or jelly-like, depending on the makeup of it. So that is the appearance of loose connective tissue. You should be familiar with the different elements that are in there, the elastic and connective fibers and the fibroblasts. Okay. All right, next is dense connective tissue, and it has two subtypes as well. So the two subtypes are regular dense connective tissue and irregular dense connective tissue. And so that's not too complicated, and they pretty much are self-descriptive. So regular dense connective tissue is shown here in the right-hand picture, where the connective tissue appears in a regular parallel bundle, okay? So it looks very straight and orderly. Okay, the irregular then, as its name implies, is all over the place, okay? Random arrangement to the fibers. This one here on the left is irregular dense connective tissue, okay? So regular dense connective tissue, you're going to find in tendons and ligaments, and the dark purple again here are the fibroblast nuclei, the cells that are present in dense connective tissue. And then in irregular dense connective tissue, which looks like this, again, we've got that random arrangement to the fibers, and you're gonna find this in the skin, in the lower levels of the skin, random arrangement of fibers. And again, you can see the fibroblasts here. Okay. All right, let's move on. Okay, so the third type of connective tissue is adipose, which of course is fat. You've got these large droplets that are adipocytes. These are our fat cells, okay? And they hold the lipid component of fat, which when the slide is prepared is actually lost. The lipid component is dissolved, but it would be in the center. And as fat is stored in these cells, the cells will swell, okay? So you're gonna actually have like a swelling as opposed to a multiplication of fat cells. It's an increase in size of the fat cells for the most part, okay? All right, let's move on to cartilage, okay? And there's two types of cartilage as well, but first, the cartilage cells themselves are called chondrocytes, okay? And there's also a lot of extracellular matrix in cartilage. Now these guys are avascular. Cartilage tissue don't have blood vessels interspersed, okay? So here's our one type of cartilage cell, cartilage tissue called hyaline cartilage. And then the other type is elastic cartilage. Hyaline cartilage is going to be found at the ends of your bones and in your respiratory passages surrounding the trachea. For instance, we're going to see hyaline cartilage. And then elastic cartilage, you're going to see in the ear and the epiglottis, which is the little flap that keeps you from choking, keeps your windpipe and your digestive tract separate, okay? And doesn't allow you to choke when you're breathing and swallowing air or swallowing food at the same time. Okay, so that little flap has a lot of elastic cartilage. That's shown here, and the hyaline cartilage is the picture on the left. There are little cavities called lacuna. This is one. This is a lacuna which hold the cells. So the chondrocytes are kind of grouped together in sets of 2 or 3, and they are in little cavities called lacunae, okay? And so that's what those are there, cells within a lacuna, okay? All right, so I'm gonna move to the next slide where you can see an example of hyaline cartilage. And like I said, that surrounds the trachea. And we have a slide that looks just like this. So this is your trachea, this is your esophagus. And if you look around the trachea, there's these like arms of tissue that surround the trachea. And if you increase the magnification of those arms, it'll look just like this picture here. Okay, so you can see this is your hyaline cartilage with your chondrocytes in a lacuna shown there. Okay. All right, let's move on to bone. All right, bone is the fifth type of connective tissue. We are going to look at a slide that has ground bone, so you can see the exposed surface of the bone, and you should be able to identify different structures within that bone tissue. Okay, so those structures we would like you to be familiar with are Haversian canals, lamella, lacuna, canaliculi, and Volkmann's canals. Okay, so let's start with the overall picture. This is our bone, our ground bone, and there's little what we call osteons or Haversian systems that make up the bone. Here is one osteon, here's another osteon. It almost looks like the rings of a tree with like a dark spot in the middle, okay, and lots of little other dark spots, which we're gonna name and describe. So this is what the bone surface would look like. The entire like ring with all of its components is called an osteon. And then the very central dark canal is the Haversian canal. Okay, this is where you're going to find a lot of blood vessels and nerves traveling in that Haversian canal. Okay, those of course are not able to be seen actually when the tissue is prepared for viewing. They're lost, but the dark canal is where they would be, the Haversian canal. Okay, and then as you might notice, there are rings of bone. There's almost layers like the rings of a tree. These are called lamella. Okay, so the rings of bone that kind of extend in concentric circles around the Haversian canal. Those are lamella. And then in the lamella, you're going to see those little dark spots shown here. Those are lacuna. That's the space that holds the osteocytes, our bone cells. Okay, so the bone cells are going to be, again, called osteocytes, and they're in lacuna, just like the chondrocytes are in cartilage. Okay, all right. And then these little fibers that extend out between the different lacuna and between the Haversian canal and the lacuna, those are called canaliculi, and they connect the different lacuna to each other. Okay. And the last term for bone that we would like you to be familiar with is a Volkmann's canal. And that is basically, it's similar to a Haversian canal, but it runs perpendicular. So it connects different Haversian canals to each other. So if you think about it, this circle here is one Haversian canal that's like running out the screen towards you. And this might be another Haversian canal running, again, in the plane that would be sort of running into the screen. Okay, and then the canal that connects those two, that's called a Volkmann's canal. Okay, transverse canals. All right, let's move to our next slide and discuss blood, which is our sixth type of connective tissue. Okay, so this is a slide with blood adhered to it. There are a couple different components to blood. Plasma is the colorless fluid that's going to be surrounding our cells. Okay, so you've got that clear section there, that's plasma. We've got red blood cells and white blood cells. Okay, so erythrocytes are the proper name for red blood cells. They're going to be the pinker, smaller cells. Okay, so all these are red blood cells. And then the white blood cells are called leukocytes, and they are larger, and they have there are different types of leukocytes, but they function in immune response and defense. Okay. And then platelets are the broken up pieces of cells, fragments, that are suspended in the plasma, and they help with clotting, okay, when an injury has occurred. All right. Third type of tissue is muscular tissue, okay? And we've got 3 different subtypes of muscle, okay? Skeletal, cardiac, and smooth. So you're probably fairly familiar with skeletal muscle to a degree anyway. It is strong, it has a quick response, and it contracts on a voluntary basis, okay? The other types of muscle are involuntary. Cardiac obviously functions without our plan in it, right? You don't have to think about it. Quick responses, involuntary contractions are characteristic of cardiac muscle. And smooth muscles are also involuntary, but they're weaker and slower contractions, okay? So let's talk about those a little bit more on the next slide. Skeletal muscle is pictured in the top 2 pictures here, left and right. And I have different orientations shown for that as well. So the top right, like the top half of the top right picture is skeletal muscle, but it's again as if that skeletal were running out the screen at you. Okay, that's the cut end of the long fibers. Okay, so that's like a transverse cut versus the longitudinal cut, which is running sort of parallel to your computer screen. Okay. All right, so but this is all skeletal muscle, the top 2 pictures. Again, that's voluntary and quick and strong contractions. As far as appearance, skeletal muscle, for you to be able to pick it out, has a lot of striations, and that is why I selected this picture on the left. Striations, cross striations, are these little lines that you can see. It almost looks like different bending of light, right? There's like these little lines that are running in this direction, even though the muscle fiber is running in a different direction, right? The opposite direction. So these little lines are called striations, and they are most evident in skeletal muscle. Okay, skeletal muscle can have different numbers of nuclei. It's not just one nucleus per muscle cell. So if you look here at this bundle, okay, you can see there's a couple different nuclei, and they are located peripherally. So along the sides, along the outside of that muscle fiber, okay? And these are long, unbranched fibers, as you can see in this here. And you can also see the peripheral nuclei here in these as well. It's not centrally, the nucleus is not centrally located, okay? All right, that's skeletal muscle. Let's move down to cardiac muscle. So this picture in the middle is cardiac muscle, and it has some striations, but they're less visible, okay? So you might see some, but the one thing that is really gonna stick out at you, hopefully, in cardiac muscle are these things called intercalated discs. And these like lines that are very dark that sort of run across the muscle fibers. Those are intercalated discs, okay? And you'll also notice that cardiac muscle has more branching fibers. So there's like a branch running off here and here. And they kind of, like I said, are more branching. And then their nucleus is centrally located. So here's a nucleus, here's a nucleus. You don't have all those peripheral nuclei like you have in skeletal muscle, okay? All right, and let's move down to smooth muscle, sometimes also called visceral muscle. We already discussed that it contracts involuntarily. There are no cross striations in smooth muscle, okay? So you're not gonna see those striations like you see in skeletal muscle. You only have one nucleus per cell, okay, and that's really evident here in the cut sections, the cut ends, where you can see you've got like this nice centrally located nucleus in those fibers. And if you look at the longitudinal section of the smooth muscle, you can see that this muscle fibers are kind of spindle shaped, meaning that they're thicker in the middle and thinner towards the sides, okay? They have like that spindle-like appearance to them, okay? And of course, we're gonna find these in our internal organs, lining the internal organs. It helps with movement of our food through our digestive tract. Also found surrounding blood vessels, okay? And they have slow, weaker, involuntary contraction, okay? All right, that's it for muscle. That was our third type of tissue. And now we are moving to nervous tissue, our fourth type of animal tissue. Okay, so nervous tissue is found in the brain and the spinal cord. Okay, it has neurons, which are the major communicating type of cell. Okay, the send signals, receive signals. And then it also has different types of cells characterized as neuroglia. Okay, so these are support. They help with defense and protecting and nourishing the neurons. Okay, but the neurons themselves are the ones we're going to look at most. So the top picture here is obviously a drawing of a neuron where you've got the cell body, which is also called the soma sometimes, soma or cell body, which holds the nucleus. And it has lots of different projections coming off of it, all these little branching fibers that come off of it, okay? And some of these are called dendrites. The dendrites receive signals from other cells, okay? So they take input in to the neuron. And then this one long one here is the axon. That helps to send the signal from this neuron to another neuron or to a muscle to convey information down the path, down the line, okay? So the cell body, we talked about, it's going to be located in the gray matter of the brain and the spinal cord. Okay, so if you've heard, sometimes the brain and the spinal cord, or the brain might be referred to as gray matter or white matter, different parts of it. The gray matter is where you're going to find the cell body, okay? And then the white matter is where you're going to find these long axons. And the reason it's called that is because axons are surrounded in what's called myelin sheath. It's a lipid-rich substance that surrounds and helps the neuron to send a signal, okay? And so that's has a white coloration to it. And so that's why the axons are present in the white matter versus the cell body being present in the gray matter, okay? Because that myelin sheath gives it the white color, okay? And then lastly, on the end of the axon are terminal or synaptic buttons. This is where the end of the line is for that neuron. And now we need to continue the signal into another cell, but first there's a space, okay, a synapse, where that terminal end of the axon releases usually chemicals into a space, okay? All right, and so that's a neuron. Types of glia include astrocytes, oligodendrocytes, and Schwann cells. Oligodendrocytes and Schwann cells are the cells that provide the myelin sheath for the axons. Okay, so they are present in the central and peripheral nervous system, respectively, and will help with conveying the signal for the neuron. Okay. All right, now the slide that we have for the nervous system is a cut through the spinal cord. The spinal cord has the gray matter on the inside. And again, the gray matter is going to be where you're going to find the cell bodies of your axons. And then the white matter for the spinal cord is on the outside of the spinal cord. This is actually flipped in the brain. You've got the white matter on the inside and the gray matter on the outside in the brain. Spinal cord, you've got gray matter on the inside, white matter on the outside. Gray matter is where your cell bodies are. White matter's projections, those axons, okay? All right, so in the middle, this gray matter, you're gonna see like this butterfly shape, characteristic of the gray matter. In the middle of that is a central canal. That's a space where you're gonna have cerebral spinal fluid traveling. And then just to orient you, this is the dorsal side and this is the ventral side to the spinal cord. So of course, dorsal again is towards the back, ventral is towards the belly. Okay, and all of this sensory information that's coming in is going to come in through the dorsal side. So this dorsal root or dorsal horn is where you're going to see sensory information. All of the motor information, so signals telling you to move your arms, telling you to walk. Motor information is going to be conveyed in the ventral side of the spinal cord. So we're going to be looking in this ventral horn right here on our slides in order to see our motor neuron cell bodies, okay? And so when I magnify that and look through our highest objective lens, you're going to see something like this lower picture, the picture on the bottom half of the screen here. Okay, so this is a motor neuron cell body, okay, where you have the nucleus, labeled A, the nucleolus, labeled B. C is, again, the cell body, or soma. The reason it has a darker appearance is because that the staining is for this substance called Nissl substance. And that's present in the neuron cell body as well as in dendrites. It will extend down in the dendrites, but it's not present, Nissl substance is not present in the axons, okay? So this is one way that we kind of distinguish between the projections coming off of this cell body. So again, cell body of a neuron has lots of projections. Some of them are axons, or some of them are dendrites, and one usually is an axon. The one that does not have staining from that Nissl substance, which it looks like, see, you can kind of see there's almost a lack of staining. It looked like there would be a projection coming here, but you don't see that staining, you see end. That indicates that this projection is the axon, whereas the other projections that do have a little bit of staining going down, those are your dendrites. Okay. All right, I think that's it. So that wraps up our talk on different types of animal tissue. There will be additional videos corresponding to the slides that you would have looked at in lab this week. Okay, so I will post those separately just so it's not too much at once. But if you have any questions, as always, you can email me at cfry@lvc.edu, and I hope y'all are staying healthy and safe and having a good time at home. All right, I hope you have a good week, and I'll talk to you later. Bye-bye.